Okay, yeah. So today I was going to talk a little bit about my work on cell segmentation and spatial transcriptomics. This sort of framework I call cell simulation as cell segmentation. To give a little background of this work, what sort of prompted it is, so I work at the Fred Hutchinson's Cancer Center. And a couple of years ago, I started working with sort of a small group within that institute called the Fred Hutch Innovation Lab, or we call it PHIL for short. And our basic mission is to try to be the designated adopter of new technologies. So one of the main new technologies we've been trying to adopt is new spatial transcriptomics platforms. And so we've been running a number of pilot projects for different groups at Fred Hutch, where we sort of run the data and take a first look at them and then sort of help them get up and running with it. So that's sort of given me a nice viewpoint to look at a lot of different types of spatial transcriptomics data. We run a lot of tumor biopsies, but we've also done, we're doing a lot of bone marrow aspirate right now, skin biopsies, TMAs, things like that. And so early on looking at this data, it was really exciting, but the thing that frustrated us most about it is that cell segmentation error seemed to introduce a lot of complexity and complication into the data. So I started to try to think about how we might improve cell segmentation in this data a little bit. To give you sort of a broad overview of what imaging-based spatial transcriptomics platforms looks like. This is a schematic showing 10x's Xenium platform. They all work in a vaguely similar manner. So this sort of works as a stand-in for a few other platforms. What's important to understand is these are probe-based platforms. So they involve, you have your sample on a slide. You're ligating probes to specific RNA targets. These probes have basically a barcode that gets decoded through multiple rounds of imaging. And so the readout from these is basically you have a fairly precise position, and then this barcode gives you what gene is at that position, basically. So like what this data kind of looks like here, this is a skin biopsy. Here on the right is what this transcript readout data looks like. So you have fairly precise XY positions, and you have a gene. Here I'm just showing 4 genes, but the gene panels, initially were a few hundred, and now they sort of pushed it to a few thousand on these. And I think they're probably eventually going to try to push this to most of the transcriptome. So you can see, just plotting the XY positions of these genes gives you some picture of sort of the dynamics of transcription throughout the sample. So you can see this KRT5, this keratin gene is expressed in the epidermis here. Lysozyme is expressed right under the epidermis because there's a lot of macrophages recruited there. You can see CD3 where there's T cells. Collagen here, there's mostly these fibroblasts in the dermis. So you can sort of pick things apart that way. And then also in these platforms, usually they support some additional imaging. So at the very least, you can do like a DAPI stain, like what's shown here on the left, which can help you sort of locate different features that don't necessarily show up as RNA. Okay, so that's basically what the data looks like. Kind of the temptation would to view this data as like single cell RNA-seq with spatial coordinates. But unfortunately, it's not quite that. And the big complication we have to treating it like that is that single cell RNA-seq has cellular barcodes, which you can have doublets and things like that, but otherwise lets you pretty precisely and unambiguously assign transcript counts to particular cells. Here, there's no cellular barcodes, so there has to be some additional kind of inference done if we want to have cell level expression estimates. And so the classic way to do this is image-based cell segmentation. So we have some additional imaging, as well as our RNA readout, and we run some sort of cell segmentation algorithm on that imaging. So these days, that's all basically deep learning algorithms that have been trained on, many hand annotated training examples. So basically, someone has to go through and draw outlines of all these cells and train it on many examples. And those are okay. And basically, what we'll get is a 2D boundary, and you can just test if those RNA molecules are in or outside a particular cell boundary. But recently, with the spatial transcriptomics data, there's been kind of an exploration of the idea of actually using the transcript data itself to try to do segmentation. And this potentially has some advantages, because here with the image-based segmentation, it's sort of done without any knowledge of the transcript data, but with the ultimate objective of assigning the transcript data. So if we actually had the transcript data, we might be able to tell something we can't quite tell from this imaging data. And that's not to mention, you can also have things like this image might not be perfectly registered to the RNA readout and things like that. So I think this method in 2022, Baser really pioneered this idea. Unfortunately, has a few problems, which we'll discuss. But this sort of piqued our interest because this seemed to improve on the segmentation results in some ways, but have some pretty significant downsides. So with that said, we could take a closer look at what exactly happens when cell segmentation is incorrect and why it's such a problem for this data. So like I said, we run a lot of tumor examples, and a lot of what we're interested in is what immune cells have infiltrated the tumor and sort of what's different about those immune cells versus immune cells that are outside of the tumor. So that's sort of like a very high level comparison that we might want to do on a lot of these samples. So if we have like a T cell that's infiltrated a tumor, so maybe it looks vaguely like this, where these dots are RNA transcripts, and we have like a T cell surrounded by tumor cells. So on the left, this is what it looks like if it's properly segmented. We get the right boundaries, so we know where all the RNA transcripts belong. We ascribe them to the correct cells. But what happens if the boundary is a little bit off? So of course, some of the transcripts get misassigned, which is not good. Of course, that introduces error into the data. But it's actually a pretty pernicious error for a few reasons. So first of all, kind of the thing we're trying to study is sort of the relationship between gene expression and proximity of various cells. So now we've pretty badly confounded those two things, because if we want to say, what's unique about these T cells that have infiltrated the tumor, well, it looks like they have a bunch of tumor genes that are upregulated. But of course, that's just a false positive result due to the missegmentation. And of course, we could ask the related problem, or the related question, what's unique about the tumor cells that are adjacent to immune cells? And they might look like they have T cell genes upregulated, which is, of course, is another false positive. So we've confounded the thing we'd like to look at. But then there's a more subtle error, which we see actually quite a bit, is that you don't get sort of equal amounts of transcripts in every cell. So tumor cells tend to transcribe a lot of stuff. And so that manifests in these spatial transcriptomics experiments as you have a lot of basically transcripts per cell in the tumor cells. Whereas a lot of immune cells, they have a much more, I guess, well-defined expression program. And so they'll have a lesser readout. And so what we see a lot is when you missegment, what tends to happen is that this tumor signal will smother the signal from this T cell. And when you're going through and like clustering things and trying to define cell types, this might just look like a tumor cell if you smother enough of that signal, this T cell. So it will also tend to hide particular subsets of cells in a systematic way. So it's a pretty bad source of error. It's not just uniformly distributed noise that's introduced. So the inspiration that I took for trying to come up with a new segmentation algorithm was this kind of interesting niche corner of the literature that I found called cellular POTS models. And the idea here was to basically build very simple simulations of cells. So if you've ever seen like Conway's Game of Life, like a sort of simple simulation on a grid where you have a few different rules and then you run it and it produces these interesting patterns. The idea is to kind of do something like that, except basically make it more realistic and kind of probabilistic. So no two runs will be exactly the same. And so the idea is, instead of having like really simple rules like Conway's Game of Life, you have kind of like an energy function, which tells you like a more or less probable configuration. And then you run this kind of sampling thing and you try to produce some interesting effects that you might observe in real life. So this is kind of the classic example from, I think the first paper about this was from '92, and they're basically demonstrating cell sorting. So basically, in this sort of simple simulation, you have two cell types, and they basically have an affinity for cells of their own type. And so even if you initialize them scrambled and you run it for a lot of iterations, they'll tend to migrate and form big blobs of the same type. So I don't need to get into too many details about how these cellular POTS models work, but the idea is pretty simple. It's a sampling thing. It's all on a big grid of pixels, or you can do it in 3D in voxels. And all they're doing on each iteration is you're sort of perturbing the boundary and then evaluating whether that kind of increases or decreases the energy function, where the energy function is just something you made up. So it could be sort of anything. And the idea is if you run this for, say, millions of iterations, you get some interesting behaviors. And so this is kind of a lot of what the cellular POTS model literature is can we come up with an interesting energy function that elicits kind of some interesting behaviors? So here's the original paper in '92 where they're doing this kind of cell sorting experiments. This is a very interesting paper in 2015 where they've done sort of this actin-inspired model to basically simulate some behaviors of cell migration. So here, I think they're trying to simulate chemotaxis. So these cells are kind of following this chemical gradient that they've simulated. Here's one from 2012 where they're basically trying to contrive something to mimic some of the features of wound healing. So it's not like a highly sophisticated, detailed simulation of cells, but it's with some simple rules, you can sort of generate some interesting examples of cell-like behavior. So I was thinking about this, and I was thinking you could take this model where you can sort of plug in anything into the energy function, and you could, instead of sort of these contrived rules to produce interesting behavior, you can plug in a probabilistic model that kind of scores the cell boundaries based on how well they explain the data that you observed. So if the cell boundaries sort of make sense for where you observe the RNA molecules, that sort of scores highly and is more likely. But if it doesn't make sense for the observed transcript distribution across space, then it scores poorly. And the idea is the same idea with the simulations. If you run it for a lot of iterations, hopefully you come up with something that plausibly explains the data. And so the basic modeling principle I'm adopted here for this is what I've been calling the bag of RNA model, which the pretty strong assumption is that RNA is just floating uniformly at random within the cell. So I'll talk about some exceptions to that rule. But if we suppose that's true, then it turns out this is fairly easy to build a likelihood model for and run this sort of simple cell simulation. And so basically, if the cell has a boundary where it looks like a uniform spatial distribution of kind of the same composition of genes, that tends to score highly. Whereas if you have something like a non-uniform mixture of genes, that tend to score poorly. So here's like a very simple toy version of what this looks like. So in this sort of contrived example I came up with, there's just 4 genes, and they're fairly cell type specific. Of course, the types are sort of simulated or contrived here too. And if I just start with nothing and kind of run this simulation with this bag of RNA model, right, these cell boundaries go all over the place, and eventually they try to gradually converge to something where it looks like, you know, solid-ish colors in a lot of these cells, or at least a decently good approximation of that. So that's really the basic idea. So then, like, the obvious question is are cells really bags of RNA? Which I think is maybe slightly controversial assertion, but I think they're mostly bags of RNA is kind of my claim. Of course, RNA localization is a thing, but I think most genes are not highly localized, and most cell types are not highly localized. And I can take a few examples here of kind of attempts at like a systematic measurement of RNA localization in cells. So here's from this 2024 paper. I've sort of replotted their data to make it clearer here, but right, x-axis is proportion of transcripts, and this is just a density plot. So you can see most transcripts are just in the cytosol, they're kind of floating around, and then you have a small proportion they're highly localized to the membrane or the nucleus. There's been some attempts to build models of subcellular localization based on spatial transcriptomics, and I think like in an unbiased fashion. And I think what they tend to find is that predominantly what you see is you know, most genes are not highly localized, and the ones that are, it's usually kind of a membrane nucleus, or I should say cytoplasm nucleus imbalance for the most part, with some membrane localization in some cases. So yeah, so the bag of RNA assumption, it's not true, but it's true enough for most cell types, I think, to serve as a fairly good heuristic for segmentation. There are some examples of cell types where kind of all of the RNA is localized in a way. A good example that I've been dealing with recently, trying to help a group with, is adipocytes, where a lot of the cell will be taken up by these lipid droplets. So like the RNA in the cell will be pushed to one side or the other. So that's a case where kind of all of the RNA is highly localized, which is problematic for this model. There's other cases like neurons and things like that, which are problematic for every model, but I think particularly have some sort of global RNA localization going on. Okay, so to see what this looks like on some real data, kind of zoomed in. This is MERScope data of a lung cancer sample. And so, yeah, so this is the segmentation result on this top panel. And then what I've done is I've kind of zoomed way in, so you can kind of see like what one step of the simulation looks like. And a single step basically is, let's try like nudging the boundary between these two cells over by 1 pixel, and then kind of evaluating the result. And in this case, we nudge the boundary down, and you have more of these purple transcripts, which are B cell specific transcripts, along with other purple transcripts. And so that will be a higher probability result, and we're likely to accept that proposal. But if it makes it worse, we tend to reject that proposal. And so basically we're running these random perturbations and accepting them probabilistically for many, many iterations until it converges to something that's high probability. So the other trick, because we've sort of built a probabilistic model, is we can extend it a lot of ways. And one of the key ways we've extended it is, I think somewhat unique to our model, is that we don't assume that the transcripts are observed with complete accuracy in the XY coordinate. So we allow for the possibility that either the readout is slightly imprecise, or transcripts have kind of migrated from where they should be, which I think can happen because there's a permeabilization step that goes on here. So the probes can get into the cell, and that can also cause some RNA transcripts to leak out of the cell and sort of be positioned not in the cell, but nearby, which sort of complicates the segmentation problem. but we're able to clean that up to a certain extent by basically repositioning transcripts if it seems like it's in an unlikely position. Okay, so evaluating this, it's evaluating segmentation quality is a difficult task. It's sort of the image-based segmentation world. What they'll tend to do is compare their prediction to some hand-drawn estimates. So basically data that someone's labeled by hand. And I feel strongly that that's the wrong criteria for a spatial transcriptomic setting, because what we should really be focused on is correct assignment of transcripts to cells. So I've tried to get at this with the spurious coexpression metric that I came up with, which is basically taking pairs of genes that we think ought not to be co-expressed, and then basically measuring their rate of co-expression. So you can see across, I've done some comparisons here. The method we came up with is called Proseg, which is short for probabilistic segmentation. So we're usually able to reduce the spurious co-expression by quite a bit across several different platforms. So Cosmix, Merscope, and Visium are some of the main ones. Visium multimodal is kind of their new segmentation add-on. So you pay extra and then have these additional stains, which we're still able to beat with this transcript-based segmentation. So that's not the only metric we should be interested in. We should also hope to assign a large proportion of the transcripts, because unless we think there's a very high rate of noise, most of those transcripts should be coming from cells. So we are achieving that here. And then we should also have a plausible number of cells. So if there's a huge disagreement between, say, like segmentation based on nuclear stains, which are shown in blue on this bottom set of plots, that would be problematic. And so you can see there tends to be an agreement on the number of cells, and the exception being Bézier here. So I mentioned us having some problems with Bézier, and that's sort of one of the main ones, is that it'll tend to wildly over-segment and produce more cells than we think there are. So yeah, so Bézier is another transcript-based method. So kind of working with transcript-based methods, the error mode of the transcript-based methods we found is kind of akin to cellular gerrymandering. So these are classic gerrymandering examples from the US. So basically, like, if you have complete freedom to draw borders, in this case of congressional boundaries, you can sort of engineer a particular statistical outcome. So if you have a statistical model and you have complete freedom over the cell boundaries, it's possible to engineer basically over parsimonious cells that are not plausible. But we think we avoid this, whereas Bézier sort of falls prey to this, in part because we sort of constrain to the nuclear segmentation to prevent introducing far more cells than there actually are. But we also have a more sophisticated probabilistic model backing the whole thing. So constraining morphology to prevent this gerrymandering problem is also a big issue that I continue to iterate on. Fortunately, there's again examples or literature to pull from the cellular POTS models. So basically, you can have sort of a prior on how kind of circular your cells are, and if you relax that prior, you can get more jagged edges. But yeah, so that's sort of one of the main constraints to prevent this sort of gerrymandering problem. So yeah, so now to show a few examples of what this looks like in real data. So first is this Merscope lung tumor example that I showed a glimpse of earlier on. So this first set of plots is, just UMaps on different segmentation methods. So I'm showing cell pose which is image-based segmentation, both looking at just nuclei and then trying to segment the entire cell. Here's Bézier, this sort of main other transcript-based method than our method Proseg. And yeah, so the UMaps are a little bit different. But one of the big, pretty consequential differences we noticed is that in trying to annotate neutrophils, we actually have dramatically more neutrophils in Proseg than when compared to, for example, Baser or CellPose. So here we had 16,000 in CellPose and 46,000 in Proseg. And so the reason we think this is sort of exactly what I described earlier, where a very high population cell can sort of smother out the signal from a low population cell. So in this particular data, neutrophils are very, very low transcript count. So this is this orange density line here, whereas tumor cells are at the other end. They're sort of the highest transcript count. So they're very high density. And so this tumor has a bunch of infiltrating neutrophils, but the signal from that has been smothered to a large extent by kind of just missegmentation and the tumor signal just being so much stronger than the neutrophil signal, especially when they're surrounded by tumor cells. Yeah, so I mean, the degree of neutrophil infiltration is kind of prognostic marker that we might be interested in, but it's sort of systematically undercounted by missegmentation, I think. So here's actually another very different lung tumor that we're looking at. In this case, this is Xenium data. Yeah, so again, I've done a few different segmentation methods. These are UMAPs at the top. We get a cleaner UMAP, which doesn't prove anything, but is suggestive, I think. And this is a very different tumor because we don't have that huge neutrophil population. We do have a big macrophage population, though, but we don't have the same issues with underestimating macrophages per se. But this is a good opportunity to look at sort of like the basic analysis that we might want to do, which is compare macrophages that infiltrated the tumor to sort of more distal macrophages. So here's like the simplest version of that sort of analysis, where let's just do pairwise differential expression between these two groups of macrophages. So here's what I did exactly that here using different segmentation methods. And these are just kind of volcano plots. So x-axis is fold change, y-axis is negative log p-value. And so these genes I've colored in red are sort of suspicious examples because they're basically genes that are highly expressed in the tumor. So if they appear upregulated in macrophages that are adjacent to tumor, that's suspicious. And what's probably going on is missegmentation, and those macrophages are picking up tumor genes. And so sure enough, we're able to not eliminate, but reduce a lot of these false positives we see in some of these other segmentation methods. So I'm going to show a little bit of proximity analysis. This is sort of the method that I came up to. There's different ways of measuring proximity, but the way I'm measuring proximity in the examples I'm going to show are based on random walks and basically measuring how many steps on average of sort of a neighborhood graph to get from one cell to another set of cells. So in Markov chain analysis, this is called the expected absorption time. But basically smaller numbers means sort of things are closer in aggregate, and larger number means they're further away. So it sort of looks like this. So this is the skin example I showed earlier, but you can take every cell and measure how close it is to T cells in general, where T cells are these black cells here, and I've colored this by this sort of random walk distance. This is the same idea below with macrophages. So that's the basic idea. So yeah, so this is the main example where we wanted to do this sort of proximity analysis on. So this is renal cell carcinoma data that we ran. We had actually 4 different tumors here. It was a pretty difficult segmentation problem. This was kind of one of the earlier data sets that really prompted us to look at segmentation closely. Because the tumor is highly infiltrated with T cells. So in this example here, blue is tumor cells, green is T cells, purple here is B cells, and so on. And so we had a lot of problem with that since we had this sort of smothering effect. T cells have fewer transcripts, they're smaller, so they're harder to separate out from the tumor signal. So yes, we have much cleaner UMAP. We're able to better separate these very distinct cell types. And then sort of like the neutrophil example, we have basically twice as many T cells we're able to recover through better segmentation than what we kind of were given with the Visium segmentation. And so this let us split up T cells into a bunch of groups and kind of measure proximity to tumor. And what came up was a somewhat consistent pattern of these CXCL13 CD8 T cells showing a greater proximity to tumor. So these negative numbers being closer to tumor. So that was sort of a population we went into this kind of interested in because it can be a marker of T cell exhaustion. So we're able to show some interesting proximity results. We also picked up this population of CCL4 positive T cells, which we know kind of less about than the CXCL13 ones. But sort of having twice as many T cells gave us a lot more power to try to pick that apart. So that's basically the segmentation method as I published it, but I've done two big iterations, which I'll talk about really briefly. So that like everything up to here is basically what I've called Proseg 1, and then I've done Proseg 2 and 3, which I've released, basically trying to keep up with how the data is progressing. So one of the first things was trying to adapt it to higher plex data. 10x came out with a 5K panel. NanoString, which is now Brooker, with their Cosmics platform, came out with a 6K platform. And Proseg wasn't really scaling to that. It worked well for like the 400 genes, but 10 times as much, it tended to break. So I spent a lot of time trying to figure that out. And eventually what I settled on is basically doing probabilistic matrix factorization. In this case, what I'm doing is partial factorization. So I basically sort the genes and take the highest expression ones and leave those as they are, and then take the lower expression ones and basically turn that into a lower rank matrix. And the trick is it does this concurrently while it's segmenting. So this is not something it has to compute ahead of time. Basically, it updates the segmentation, then updates this factorization and back and forth. So that ended up working pretty well, and we're able to adapt it to basically arbitrarily high gene panel sizes. So we're able to segment the CDM 5K data really nicely, get much cleaner cell types, and end up assigning more transcripts. Using a higher, there's some downsides to using a higher plex panel, but this also let us find a pretty interesting gene. Of course, we're interested in these T cells that were infiltrating the tumors. So here's our CXCL13 gene that we're pretty interested in. But this other gene that wasn't exactly on our radar, TNFR SF9, actually popped up with maybe even a larger effect size. There's a little bit of literature about that, but not a lot. So that was interesting. So that's Proseg 2. And then sort of the more recent development is what I've called Proseg 3, which was trying to adapt the same model to, which so far has been entirely image-based spatial transcriptomics platforms, and kind of adapt it to sequencing-based platforms, which there are a few of. And the main sort of constraint with the sequencing-based ones, which are doing like something that's more close to single-cell RNA-seq, except that you're basically assigning spatial probes one way or another. And so the big difference between this and sort of the imaging-based platforms is that the spatial measurements are much more imprecise. So the highest resolution for these are like a few microns, whereas the Visium readout is at least thought to be very precise in the XY coordinate. And then the other big difference is that, as tends to be the case with, sequencing versus probe-based methods, is the data is less sparse here. So we have this transcript repositioning scheme that sort of lets us do a better job of accounting for the readout being imprecise. The data being less sparse is not inherently a problem, but it just required a lot of work optimizing the method to get it to sort of scale and run fast enough. So I basically rewrote a big chunk of this and made it a lot faster, which lets it scale to that. And that was sort of the main change that's needed. And so this is something I'm still working on benchmarking, but we seem to be making some improvements over sort of the existing segmentation solutions. So this is some Visium HD data, which is kind of one of the main platforms. So yeah, so just to talk really briefly about where I see this headed. I think this is a pretty promising direction for cell segmentation for transcriptomics data. And there's a lot of different ways you could build upon this, which I'm sort of exploring now. One, which I think will be fairly straightforward, is to do supervised segmentation. So if you happen to have like a single cell RNA-seq that closely matches your data, you can sort of train the segmentation model on that to sort of inform it ahead of time which gene should be co-expressed. There's also literature with CPMs that have basically modeled the existence of a nucleus, which might improve our results here, because I think one of the main forms of RNA localization is basically cytoplasm nuclear imbalance. Then the other thing I think is an interesting direction, which there's some groups working on this now. I've cited this Resolve VI paper, is basically trying to do a downstream analysis that kind of takes into account missegmentation. So a lot of what people use now are basically tools that were designed for single cell RNA-seq analysis. But I think if you can write tools that, like this, sort of has like a term in this regression that's trying to account for the possibility of diffusion due to missegmentation, I think that could be a promising direction. Yeah, so basically segmentation error is a huge issue in this data because it kind of confounds the thing we want to look at. We think we've taken a pretty big step in improving the situation, but we definitely haven't solved it. It continues to be something you have to be sort of vigilant about. And I think ultimately the solution will involve some more specialized downstream tools. And yeah, so we have this paper out a couple months ago in Nature Methods where you can sort of read more about the basic algorithm. And then the tool itself is also open source, so you can run it on something if you'd like.